Hello and Tashdil everyone. Welcome to class 12 biology. In this video, we will discuss about the Human Genome Project. So we will see first what is Human Genome Project. Then we will discuss about what are the methods involved in the Human Genome Project. Then what are the salient features of human genome. And finally, we will discuss about the applications of the findings of the Human Genome Project. Before we start discussing about the Human Genome Project, we need to understand what does the term genome mean. Right? So what is the definition for genome? A genome is an organism's complete set of DNA, including all of its genes. I found this definition given in the website of National Institute of Health. So a genome is an organism's complete set of DNA, including all of its genes. Each genome contains all the information necessary for building up and maintaining that particular organism. So for example, if we take ourselves as an example, a copy of an entire genome, which is about more than 3 billion DNA base pairs, it is contained in all the cells that have got nucleus. So entire genome set will be present within each and every cell within our body that has got nucleus within it. Right? So those cells which do not have nucleus, like RBCs, they won't contain the genome since they don't have nucleus. But all the other cells, rest of the body cells that have got nucleus, that they have got an entire genome within it, right? So that is the definition of human genome. And simply you can write down a genome is an organism's complete set of DNA, including all of its genes. So now, what is Human Genome Project, or HGP in short? The Human Genome Project was an international research effort. It was an international research effort to determine the sequence of the human genome and identify the genes that it contains. Right? What they were trying to do was, since we have got about 3 billion base pairs, the goal of the Human Genome Project was to sequence that 3 billion base pairs and identify what are the genes that are contained within that particular 3 billion base pair long DNA. That was the goal, right? And the project was coordinated by the National Institute of Health and the US Department of Energy. So both of these institute and department are present within the United States. But since it is an international effort, other countries were also involved in that this particular project. Those countries were United Kingdom, Japan, France, Germany, China, and other countries. Right? So therefore, it is called as an international research effort. Now, certain estimations were made regarding this project, right, before the project started. Some estimations were made by the scientists. The scientists, they said it will take about 13 years for the project to complete. For sequencing of the human genome, it will take about 13 years. That was their estimation. And then it will cost about $9 billion, $9 billion US dollars for the project to complete. Now this 9 billion United States dollars, right? This they deduced, or they got it from assuming that if it cost about $3 for each base pair to sequence, then if you multiply that $3 with 3 billion DNA base pairs, then it will be about $9 billion United States dollars. So that is the cost and the time estimation of the project. Now, since it was going to take the efforts of lots of these countries and institutes, and since it was going to take a long time, and since it was going to take about 9 billion US dollars, that's a lot of money, this project was called as mega project. This project is also called as a mega project. Mega here means huge, right? A very large project. Now, what are the goals of Human Genome Project? What are the goals? So the first goal and the second goal I've already told you before, it is to sequence the entire human DNA, which is about 3 billion base pair long, and identify all the genes that are present within it, right? So genes, about 20 to 25,000 genes. The third goal is, after sequencing and identifying the genes, you will get a lot of data out of it, right? You will get a lot of data. So one of the goals was to develop some technology through which they can store the information in databases, large databases, and to improve the tools for data analysis. After you get the data, you have to analyze the data, right? So improve tools or softwares, right, or programs to analyze the data. So these were the two fields which developed from Human Genome Project. So now the branch of science which involves the storing of information in databases and data analysis, which pertains to biology, that is called as bioinformatics. So bioinformatics stemmed out from Human Genome Project. The fifth goal is transfer the related technologies to other sectors. So whatever technologies that develops from this particular project, throughout this particular project, various technologies will develop, since lots of money were being spent. They wanted to transfer that technology to other sectors or other industries. Right? That was one goal. And finally, they wanted to address the ethical, legal, and social issues, LC, right? Short for LC. This word is important. LC, which means ethical, legal, and social issues. E for ethical, L for legal, SI for social issues. Now, since this is going to be a huge project, right, and it deals with sensitive issue of human genome, DNA sequencing, gene mapping, everything. So they foresaw that there will be lots of legal issues, ethical issues, and social issues pertaining to the project. So they wanted to address those issues, right? So they spent a considerable amount of money for addressing the issues which can come along with the project. So these are the goals of Human Genome Project. You may want to copy these down, right, and keep them ready for your exam. And I told you that this particular ELSI, the short form abbreviated one, you need to know the expanded version, ethical, legal, and social issues for LC. Okay. Now, apart from that, they wanted to sequence the genomes of other organisms as well, right? Primary goal is to sequence the human genome. Along with that, they also wanted to sequence the genomes of some other medically important, biologically important organisms like fruit fly, Drosophila melanogaster. They also wanted to sequence the genome of mouse, yeast, and other bacteria and other organisms. Why they wanted to do that was they wanted to compare the genes of human with genes of other organisms so as to draw some parallel between the two, right? So these are the goals of Human Genome Project. Now, why perform Human Genome Project, right? Why sequence the entire human genome, and why sequence the genome of other organisms? So the reason behind that particular project was to understand the effects of DNA variations among the individuals, right? If we have got two individuals, and if we compare the two individuals, they don't look alike, what makes them look different, right? What are the differences in their DNA or their genetic makeup which makes them differ from each other, right? So we can understand that, but we can understand that thing only after we know the genome of the two individuals, right? So scientists, they wanted to understand the effects of DNA variations among the individuals, right? So that is one reason. And other reason will be, if we know the genome of individuals, what we can do is we can find new ways to diagnose, treat, and prevent human genetic disorders. We have studied about genetic disorders in our previous videos. So if you want to diagnose or treat or prevent genetic disorders, you need to know the genome of the individuals, right? If there is some genetic disease within me, I have to go and sequence my entire genome, then only I will find out whether I have got that particular disorder within me or not, and what are the chances of me passing my genetic disorder to my progenies, right? So you can prevent the genetic disorders to be transferred to the progeny, right? So that is another objective that is in medical field. And apart from that, this particular Human Genome Project, it has got applications in healthcare, agriculture, energy production, and environmental remediation as well, right? So that is why scientists, they wanted to carry out the Human Genome Project. Now let us try to understand the methodologies involved in the Human Genome Project. What are the methods involved in Human Genome Project? So there were two approaches. There were two groups of scientists. One group, they wanted to do express sequence tags. expressed sequence tags, or ESTs for short. So in this particular approach, what scientists they wanted to do was they wanted to identify all the genes that are expressed as RNAs and sequence them, right? So what they wanted to do was they wanted to identify all the genes first, and then they wanted to sequence them. That means they wanted to sequence only the coding portion of the DNA. Another approach was sequence annotation. The second approach was called as sequence annotation. In this particular approach, what scientists they were trying to do, what they proposed was, they said, let us sequence the whole set of genome that are contained, right, that are contained within the human cell, and let us sequence the entire DNA, whether it is coding or non-coding, whether it codes for protein or not code for protein. They should sequence the entire DNA. So that was the second approach. So here we can see that if this is a chromosome, it is made up of DNA. In the DNA there are many regions, right? Coding regions, the red regions, these are the genes, and these are the non-coding DNA part. What the scientists who proposed the express sequence tags, what they were proposing was they wanted to sequence only the portion which are shown as red over here, genes. They wanted to identify the genes first and then sequence them and not bother about the non-coding DNA. But other group of scientists who proposed the sequence annotation approach, they wanted to sequence the entire DNA irrespective of whether it was coding or non-coding. Right. And out of these two approaches, later on the scientists they decided to follow this particular approach, sequence annotation approach, right, in which they are going to sequence the entire DNA irrespective of whether it is coding region or non-coding region, right. So sequence annotation was the approach which was adopted by the scientists involved in Human Genome Project. Now let us look at the steps involved in this particular method or in this particular approach. In the first step, what scientists have to do is they have to isolate the DNA from the cell. So let us say we have a human cell. We have to isolate the DNA. We know that the DNA is present within the nucleus, and we have to take the DNA out in its pure form, right? So in one of your practical experiments, you have to perform this particular thing, in which first, in order to get the DNA in its pure form, you have to remove the cell membrane. Then you have to break open the nuclear membrane, and then you have to take out the chromosomes or chromatin, the DNA which is associated with histone proteins, and then you have to remove the proteins to get pure DNA or nucleic acid, right? So that was the first step. So the first step in Human Genome Project, right, is to isolate the DNA from the human cell. After isolating the DNA, in the second step, what the scientists they have to do is they have to cut the DNA into random fragment. And we know that DNA is a very long biomolecule. It's a very large biomolecule, right? So we have this particular long DNA. In the second step, after isolation of this DNA from the cell, the scientists they have to cut the DNA into small random fragments. Right? So it is easier for the scientists to sequence these small fragments rather than sequencing the entire DNA in one go. So there were technological limitations which prevented them from sequencing the entire DNA in one go. What they had to do was they have to cut the DNA into small fragments, and then they have to sequence these small fragments individually and separately. Okay, so this was the second step. In the third step, they have to clone the DNA fragments by using suitable host and by using specialized vectors, right? They have to amplify, or they have to clone or increase the number of DNA fragments so they can sequence them later on. So in the cloning method, what they have to do is, after taking out the DNA from the human cellular nucleus, they have to cut the DNA into small fragment, and then by using vectors, specialized vectors, they have to insert that into suitable host, right? And when the host divides, along with the host, along with the host and along with the vector, the DNA fragment will also increase in number, right? So that is called as the cloning method. And in this particular cloning method, we can see that the scientists they used a specialized vector with BAC and YAC. They used BAC and YAC. These two words are important, you have to know. BAC stands for bacterial artificial chromosome and YAC stands for yeast artificial chromosome. So these were the two special vectors. And for the host cell, they used bacteria or yeast. So since bacteria and yeast can multiply really quickly, so if you insert the DNA fragment within the cell, along with the cell, the DNA fragment will also increase in number. So that is called as the cloning method. And here the host is bacteria or yeast, and the vector is bacterial artificial chromosome and yeast artificial chromosome. So this is the cloning part. After cloning the vector, after cloning the DNA fragments, what you have to do is you have to sequence the DNA using automated DNA sequencers, right? So they used machines which are automated DNA sequencers. They can automatically sequence the DNA, the small DNA fragments which are put into this particular machine. They will sequence it, and this particular machine will sequence the DNA according to the principle proposed by Frederick Sanger, right? So in higher classes you will learn about Sanger's method of DNA sequencing, right? So this machine worked on the principle of DNA sequencing principle proposed by Frederick Sanger, and Frederick Sanger was also the first person to propose the method of sequencing protein as well. Okay, so that thing. So in the fifth and the final step, it involved alignment of DNA sequences. So this was done with the help of specialized computer programs, right? So they fed the data after the DNA sequencer machine sequenced the DNA. They put this data into these computers, and these computers had specialized computer programs which arranged the DNA sequences, the short DNA sequences, in such a way that they can form one complete DNA sequence, right? One complete DNA sequence. That is called alignment and arrangement of DNA sequences. So after this was done, after the DNA sequences were arranged, later on these DNA sequences were annotated, right? And after that they were assigned to each chromosome. So these were the steps involved in Human Genome Project. Now the chromosome number 1, over here, this is chromosome number 1 out of 23 pairs. This is the chromosome number 1. Chromosome number 1 was the last one of the 24 chromosomes to be completely sequenced. And the sequencing of chromosome number 1 was completed in May of 2006. Now I told you before that the duration of the Human Genome Project was for 13 years. They estimated it will take about 13 years to complete, starting from 1990 to 2003. Even though they declared that the Human Genome Project was over in 2003, but the chromosome number 1 was not completely sequenced. The complete sequencing of chromosome number 1 happened in May of 2006. That is 3 years later. Okay, so that is one extra information that you need to keep in mind. Now let us look at the salient features of Human Genome. Right. The findings of Human Genome Project is the human genome has been entirely sequenced now. So what are the salient features of human genome? The salient features of human genome is the human genome contains about 3,164.7 million nucleotide bases, and in this number of nucleotide bases there are 30,000 genes, right? And if you compare, 99.9% of bases are same in all the people, right? I have got 3,164.7 million nucleotide bases in my genome, and you also have the same, and 99.9% of the sequence are same between you and me, right? So that is the salient feature, or that is the first salient feature of human genome. The second salient feature of human genome is the average human gene. The average human gene contains about 30 bases in them. So if we have a gene, right, average, if you take an average, in a human gene there are 3,000 bases in it, right? There are about 3,000 bases. I'm sorry, it is 3,000 bases in one gene. But that is average. But we have genes which contains more than 3,000 bases, and we have genes which contains less than 3,000 bases. So for example, if we take the largest human gene, which is dystrophin, so the largest human gene is dystrophin gene, and that is the largest, and it contains about 2.4 million bases, right? It contains about 2.4 million bases in it. So that is the second salient feature. The third one is the function of 50% of the discovered genes were unknown. They discovered 30,000 human genes, right? So out of them, 50% were, the function of the 50% were unknown. And less than 2% of the genome codes for protein, right? If we have an entire DNA length, I showed you before, right? We have coding sequences and non-coding sequences. So among the entire DNA, the coding part, the genome which codes for proteins, it contains only 2% of that. Okay, so that is the fourth. The fifth is, so the other parts, right, the ones which do not code for anything, those are repetitive sequences, right? So the large portion of human genome is repetitive sequences, right? Sequences which repeats again and again, right? So those are called as repetitive sequences, and these repetitive sequences are quite important, right? Even though they don't have any particular function, they don't code for mRNA, they don't code for any protein, but they have got important function in the stability of the DNA and in evolution as well, because they help in preventing unnecessary mutations, right? So that is one. And some extra information that people found from Human Genome Project was that chromosome number 1 has most gene. I told you before, right? It has about 2,968 genes. Chromosome number 1 has 2,968 genes, most number of genes on a chromosome. And chromosome Y, the Y chromosome, the one which is present, the sex chromosome present in boys, males, human males, the Y chromosome has the fewest gene. On a Y chromosome, there are only 231 genes present on it. And finally, the salient feature of human genome is the scientists, they identified about 1.4 million locations where single base DNA differences occur in humans. So these single base differences, they are also called as SNPs, SNPs, right? Pronounced as SNPs, and SNPs stand for single nucleotide polymorphism. Okay, so these are the salient features of human genome. Now finally we will discuss about the applications of human genome. I have already told you that human genomes have got Human Genome Project will be applicable. The findings of Human Genome Project will be applicable, will be applied in finding new ways to diagnose, treat and prevent human genetic disorders. I already told you that before. Apart from that, what now scientists they expect as new approach to biological research will come up, right? New approach or in biological research will come up. Earlier the scientists they used to work with only one gene at a time. If I am a scientist, I have to work with one gene, right? Before there was limitations like that. I cannot work with other genes because that will take up another time. That will take up my time, right? So now what scientists they can do after the completion of human genome and from the findings of Human Genome Project, scientists they can now work on all the genes of a cell, right, simultaneously to understand how they work together, how the genes they work together in an interconnected manner, right, in an interdependent manner to keep the cells alive or to keep the biological cell functioning, right? So that is one application which you have to keep in mind, right? So what is the use of sequencing the entire genome of an individual organism is, if you are performing a research on that particular organism, now you have the freedom to work with as many genes as you want, right? To see how those genes, that many genes, they are working in an interconnected manner to keep that particular cell alive, right? So that is the application of the Human Genome Project. Now in the next video, which is the final part of the chapter, we will discuss about the DNA fingerprinting, right? So please read about this particular section before you watch the next video. Thank you.